Way, but I do have this system. I do have this, got it. I do have this system in place where this is the lone student that showed up after I invited my morning class to come. But if I go to a whiteboard, I can show you what I'm doing on that. When we get to a discussion, I'll just sit down here and we can talk. But for now, I'm just showing a system where you can see what's being projected behind me. And I know you're all here to hear about the bloom this summer, and I'm not even going to talk about that. I've been asked to talk about changes in the oceanography of the Gulf of Maine and how they may be related to unusual blooms that we've been seeing. So let's jump right into it. Wrong computer. So the spring diatom bloom is the most important feature that people noticed about the Gulf of Maine years ago. And of course, Henry Bigelow wrote about it. I'm not going to read you his quote here, but I always wondered what he would think if he could have seen these satellite images of chlorophyll in the Gulf of Maine back when he was studying the bloom. And as you can see that I'm showing here, it's a rich diatom bloom, but there are lots of other groups of phytoplankton in there. So, of course, we want to know about monospecific blooms and some examples we've seen. Well, of course, last summer was the Tripos bloom. I really didn't get to look at it myself. I didn't get out on the water. There's been just this exchange of information sort of ad hoc with this email, but we have seen them before. This is an image that I took, I think back in 2006, where there was apparently a monospecific bloom of what we then called serratium. And I didn't compute any densities. We were out there studying Alexandrium, and so this was just a nuisance alga that got in the way. But what about other examples? We have seen examples of monospecific blooms, and so I'm just going to go through them. The one that I first got interested in many years ago was coccolithophore blooms. This was a rare event that we saw in the late 80s, early 90s in the Gulf of Maine. It showed up in visible satellite images as chalky water, and it was just jam-packed with these coccolithophores, and a sediment trap that we put in the bottom collected these aggregations of copepod fecal pellets that looked like they'd been taking Maalox and just raining down this calcium carbonate to the bottom. So the question is, what caused these blooms? And we did do a hydrographic survey in and out of the bloom area for a couple of years, and we came up with some ideas of what they were related to. And it turned out that if you look at TS diagrams of where we saw the bloom and where we didn't, and looked at the slope of those TS diagrams above main intermediate water, in other words, the near surface waters, it indicated that the waters where we saw euxii tended to be low nutrient, low phosphate, and even low iron waters. We didn't measure iron, but based on the location of these blooms, we assumed that these were waters that were not in contact with the bottom and subject to enrichment in iron. But that's about all we could tell. Then, of course, for years we studied Alexandrium. Then it was Alexandrium fundiense and then Alexandrium catanella. And we weren't really seeing monospecific blooms. We were seeing blooms where Alexandrium dominated the plankton, but they weren't really by themselves much of the time. And over the years, we put together pieces of the puzzle and came up with ideas of why they were occurring where they were. And it seemed to be related to the nutrient field, especially areas where the nitrate remained fairly concentrated after the spring bloom, which was limited by silicate, and any remaining nitrate was available for species succession to non-diatoms. We also saw what we thought was a allelopathic interference between Alexandrium and diatoms. And you can see them here. Here's Alexandrium with also some serratium back then. But notice these diatoms that are around it are very thin frustules and apparently suffering from being around them. So we saw both Alexandrium in mixed cultures with other dinoflagellates like serratium and other species, as you can see in the two green circled photographs on the right. But we also saw them with diatoms, but never with healthy populations of diatoms. And it looks like there was an allelopathic interference, not Alexandrium on diatoms, but diatoms on Alexandrium. The presence of diatoms in nearshore waters rich in silicate precluded Alexandrium. They tended only to grow offshore. So that was one of the other groups we saw that was blooming as a single species or manifest as a monospecific. The other is Phaeocystis blooms here in the Gulf of Maine. They actually were quite common at one time. And I can remember a period in the early 90s when it made headline news in the Boston Globe and of what was called the slime tide. And fishermen were reporting that their nets were coming back filled with gelatinous material. They didn't know what it was. And it turned out that it was these promesiophytes, these tiny phytoplankton that live in this gelatinous matrix. And they tend to certainly clog phytoplankton nets, but even fish nets, it turned out. I never really heard what the cause of those blooms might have been, although some work done with Zhang at UMass Boston began to, the host has spotlighted your video. Anyway, some inroads were made. It seemed that they fluctuated out of phase with diatoms, high diatoms, low phaeocystis, high phaeocystis, low diatoms. That's the third one. So we also saw monospecific blooms of Prorocentrum, especially in nearshore waters in the Gulf of Maine. This is a photograph, a couple of photographs taken by the late Maureen Keller at Bigelow Lab of some waters, and she just dipped her hand off the dock at Bigelow, and you can see it looked like she's been playing in a mud puddle. The densities of this species were so high. Now, these are supposedly harmless phytoplankton, and we never did determine what caused it and whether they were in any way harmful. Another one that I was unaware of, although I should have been aware of it and wasn't, were these blooms of what we used to call Mesodynium rubrum and now call Myrionicta rubrum, which is actually a protozoan. And one day I was sitting in my office here at the university, and a graduate student came in with a Coca-Cola polycarbonate bottle of water and said that it was just collected in Sorrento Harbor here on the Maine coast in a red tide. And it was near a aquaculture site with our salmon pen, or no, it was a cod pen, and the fish were acting funny. And so I didn't think much of it, and I took a look at it the next morning, and it was filled with these Myrionicta. And it was an explanation for what I'd been hearing for decades of red water throughout the Gulf of Maine. And I never have, in the entire time that we studied red tides or PSP and Alexandrian blooms with Don Anderson's group and Dennis McGillicuddy at Woods Hole, I'd never seen red water. And I always blamed it on the fact that I'm a little red-green colorblind. And so, well, I must have seen it, I just didn't know what I was looking at. But no, that's not the case. And this is a case, I hope you can see it in the projection. I can see it on the monitor that I'm looking at, where there's this streak of red water running through Frenchman Bay in eastern Maine, just adjacent to Mount Desert Island, that is just chock-a-block full of these Myrionicta, which may be an explanation of all of these occurrences that we have heard about of red water and red tides in the Gulf of Maine. And it's actually this beast and not Alexandria, especially the preponderance of observations in Frenchman Bay. Never did figure out what was causing it. It's a protozoan. It was ingesting phytoplankton and using them symbiotically. And then I came upon this paper written in 1932, which was a bit of an embarrassment because this guy noticed, if you read here, great patches and lanes of red water, swarms of small red animals, and it turned the water from a brick to a blood red. And then I started looking more at the literature, and one of my advisors in graduate school, Bernie McAllis, actually wrote a paper on this as well. And of course, I ignored it. It wasn't in my field at the time in graduate school, but this too has been well known. So there are occurrences of these rare, unusual monospecific blooms of phytoplankton that have gone unnoticed in the past. And this one last summer may have gone unnoticed if it had not been so widespread, which made it unusual. So therefore, we have observed these in the past, certainly Tripos last summer, or Serratian Tripos, whatever we're calling it. And the question I'm asking, are there any clues to what could be behind these monospecific blooms in some of the changes we've been seeing in the oceanography of the Gulf of Maine and neighboring waters? And so I'm just going to jump quickly into a summary of Gulf of Maine water masses and nutrients and look at some of the changes we've seen in the past 15 or 20 years, where we see an alternation of dominant water masses in the Gulf of Maine and changing nutrient loads as a result, and also shed a little bit more light on this warming of the waters of the Gulf of Maine, which we've been hearing about for quite some time. First of all, to put it into context, the Gulf of Maine sits in a dynamic region where cold and warm ocean currents meet and mix just offshore in what we call the slope sea. We have cold water coming down in the Labrador Current, both on the shelf and offshore, and the warm Gulf Stream moving up, heading offshore at Cape Hatteras, spinning off eddies and rings here. And this part of between the edge of the continental shelf and the Gulf Stream is known as the slope sea, where these waters meet and mix. Oops, wrong computer. And so here's a summary of the current systems we deduced were responsible. It's a pretty small figure, I know, but By the way, I'm going to be showing a lot of information with a lot of detail and slides. And I've told Sally that I sent her a copy of this PowerPoint, and they are free to send it to, or you can also just send me an email. I'm happy to give you a copy of this PowerPoint. There's nothing proprietary here. I use it in teaching. Students use them all the time. So feel free to use any information, any figures, whatever, but at least you'll be able to read better some of the references that I cite when I show these things. But anyway, here's a depiction of these cold water currents coming down both on the shelf and along the slope, the Gulf Stream moving north, and these two water masses of Labrador slope water, which is cold and fresh, and warm slope water shed off the Gulf Stream, which is warm and salty. And they meet and mix in the slope C. And depending on whichever water mass is dominant, near the mouth of the Northeast Channel is what enters into the Gulf of Maine and tends to drive the nutrient loads that are in the Gulf. And so we have the cold, relatively fresh, but denser Labrador slope water, which tends to come in along the bottom, as well as Nova Scotian shelf water that comes in at the surface. And we have warm and salty slope water from the south that comes in above Labrador slope water. So here they are diagrammatically coming in, but the slope waters then spill into the Gulf of Maine through the Northeast Channel, and along with the shelf waters circulate around the Gulf. So what happens is, if we look solely at, well, here, this just shows the two major water masses, the shallow Scotian shelf water and the deep slope waters of both types coming into the Gulf of Maine, one through the Northeast Channel, one around Southwest Nova Scotia. And the Scotian shelf water is clear as a bell. In the temperature images that we get from satellites, you can see it coming in around the corner. Less visible, of course, is the slope water that comes in along the bottom, which has been known since Henry Bigelow back in 1927. You can see it coming in, hugging to the right, responding to the Earth's rotation. And it's that deep water that's the source of salt in the Gulf of Maine and also the nutrients. Now, whichever slope water comes in is very important to the nutrient loads. This is a transect taken off the coast of Labrador, going out some 400 kilometers and down to 1,000 meters, and it shows the concentrations of nitrate. And I just want you to remember these numbers, that 15 to 17 micromole in the vicinity of depths that could come through the Northeast Channel of nitrate, and silicate is around 9. That's in Labrador slope water. And warm slope water, a huge difference. Here we have up to greater than 20 micromoles of nitrate, up 10 to 14 micromoles of silicate. And so very big differences in the nutrient loads of these two water masses, whichever one comes in. So the warm slope water has 7 micromoles more nitrate than Labrador slope water, but both of them have more nitrate than silicate. So that then when whatever the source, when they come into the Gulf of Maine, silicate is more likely to limit diatom blooms first. Depending on how much nitrate is left over, that will determine which species follow in succession from the diatom bloom, which form of dinoflagellates or other phytoplankton species. Anyway, as those waters come into the Gulf of Maine, they circulate with the residual circulation, and a lot of them move up into the eastern Gulf of Maine, where the tides mix them up. They come down into the west, and you can see this river of high nutrient water in all of these surveys that we've run over the years. Then something changed. The whole Gulf of Maine changed sometime between 2008 and 2009, as this Gonzalez-Nitos paper showed, where we see changes in the Gulf Stream, where both satellites and in situ measurements showed that in about 2008, the Gulf Stream apparently migrated closer to the tail of the Grand Banks. It moved north, and that's a shift that's persisted ever since. And what that did is shown diagrammatically here, where prior to the period 2000 to 2008 of his study, I've got the Gulf Stream flowing happily here south of where it needs to be. And so we have Labrador current water coming along the shelf edge in the slope waters here. And much of it gets eaten up by the Gulf Stream, but a lot of it comes in along the slope, and that becomes Labrador slope water. It also comes in as a shelf flow across here, some going in the Gulf of St. Lawrence, some getting a spill of fresh water with it, and comes in as a surface flow as well. The warm slope water is sitting here just north of the Gulf Stream, and they mix across this boundary between them and make one or the other more available for entry through the Gulf Stream, through the Northeast Channel. Whereas after 2008, then the Gulf Stream has moved north. The Labrador current now is pinched off and is not allowed to come around the tail of the Grand Banks, or at least very little of it is, such that most of the water we see in here is warm slope water. Not only that, but That means that we also, the one result is we have a smaller slope sea. And part and parcel of the Gulf Stream changing its trajectory further north, we've also seen, or I haven't seen, others have seen an increase in the number of warm core Gulf Stream rings, some doubling of the number in the last 20 years. That brings more of that Gulf Stream water and Gulf Stream ring water closer to the Northeast Channel, where it can be mixed in into the Gulf of Maine with that warm slope water. The key element to that is that with that Gulf Stream water is very low nutrients. So a lot of slope water comes in with a mixture of Gulf Stream water and could be lower than nutrients. The data, there aren't enough data really to draw any conclusions. Now about warming in the Gulf of Maine, of course, these are the two papers that started that drew attention to the warming in the Gulf of Maine by Kathy Mills and Andy Pershing in 2013, 2015. And one is showing a little less data than the other, but a couple of things to notice. First of all, this is what they spoke of. This is the rapid warming that drew everyone's attention between whenever 2005. Both plots show it here. Curiously, though, notice it get cooler just before that. But more importantly, notice that there's an oscillation here, an apparent coherent oscillation of water masses that the authors didn't really discuss. And we've discussed it in the paper we just published that seems to be related to alternations in the frequency of Gulf Stream rings, and position of the Gulf Stream seems to be related to that. But that warming was, well, anyway, looking more closely at that warming, this is a study we just finished, and I could send anybody who wants it. Well, this is freely available online. It's an open access journal where we've taken the entire history of buoy data in Jordan Basin and put it together to show how deep water temperature and salinity have alternated in the past. And then here's the warming event here that we see in the deep waters. The gray are 200 meters, the red is 250 meters. And same thing happened with salinity. You can see that not only was there a warming of waters, but it was an increase in salinity as well. And if we do depth averaged vertically integrated temperatures and plot them both from the surface to 280 meters and then 75 meters to 280, which is to the bottom, they pretty much plot on top of one another, meaning that what we're seeing happening in the Gulf of Maine is a major bulk water mass in waters deeper than 75 meters all the way to the bottom. That something happened where at the same time we saw a change in the position of the Gulf Stream, now we see warmer water. And here are some of the, what are we calling those? Ocean heat waves that you can see showing up there, but they're not just ocean heat waves. Here are the salinities, and they're also ocean salt waves. So it isn't that the sun is shining brighter and the Gulf of Maine is warming because of some artifact of solar insolation, but it's a change in water masses coming in. And this is verified with the entire history we have of TS diagrams to show that the early years are in the cooler X's there, and the later years in the warmer colors, the red colors. And you can see that there's been both an increase in both temperature and salinity over time. And the character of Gulf of Maine waters has shifted to a warm and salty. And the real question is how much Gulf Stream water is associated with that. And that's important because, again, looking at that same transect I showed you going into the Gulf Stream, but looking at temperature, salinity, in addition to nitrate, I called your attention to the high concentration of nitrate offshore of the Northeast Channel. But once that Gulf Stream ring, which is, this is the border of the Gulf Stream here, this is the Gulf Stream. On the other side of it, notice that the nutrient levels, the nitrate levels are only 2 or 3 micromoles versus 20 micromoles on the other side. So these rings will come in and mix their warm water with warm slope water, but at the same time, dilute their nutrient load. So what's going on to drive these monospecific blooms? Who knows? My colleague upstairs constantly comes down with a hammer and whacks me over the head and said, Tom, you got to stop thinking about bottom up. You got to think about top down. Well, in the case of Tripos, I always heard, and everything I read and saw in studying plankton in the Gulf of Maine for 40 years, that nothing eats it. So there is no top down for Tripos blooms. And so light and nutrients and physical mixing properties have to be at the root of this. And it needs a lot of attention and a lot more close study. Sorry to drone on for more than 20 minutes, and I understand we'll have questions at the end. I'm done.
Unmuted. Thank you very much, Dr. Townsend. Really, really interesting stuff. Big picture trends in the Gulf of Maine. Some really interesting questions. He admitted that he didn't know the cause, so it's open for debate. All right, so our next speaker I'd like to welcome from Woods Hole, Rune Pathare. She's a research assistant from Woods Hole, and she manages instrumentation and sampling programs for harmful algal blooms research. Some of her current projects include deploying image flow cytobots on board research cruises in Alaska and the Canadian Atlantic. And she's going to be sharing with us some of the information that her lab picked up during this bloom during the past year. So I will now bring her talk up here. We can do this. Here we go. Get you now your first. I think you are all set. Take it away, Rune.
I am on a word presentation. Hi, I'm Rune Pathare. I'm here to present on behalf of Mike Brosnan, who's going to be here today. He is the PI of our lab, and I do manage all of the instrumentation for him. I wanted to share a lot of things that we have done last year. It was a really big year for us. We deployed a lot of really interesting instruments, and Tripos was just kind of an unexpected thing that we picked up last year. So I'm going to try and rush through a bunch of my presentation to get to what all of you are interested in Tripos. I also want to point out that the background of this is so much more relevant than I thought that it would be. That is the sea surface temperature anomaly. And I did not expect it to be this relevant to the talk. I just thought it was like such a cool background to have. So this is just the outline of my talk. I'm going to show you some of the Havon Northeast Imaging Flow Cytobot Network things that we've been doing, share a few summaries of our 2023 deployments, and then get to the Tripos muelleri detection in the Gulf of Maine, and use that as a case study to show you some of the image classified development that we've been doing, and the HAVOP data portal, which is a website that we built specially to show data like this. And we built a special website for Tripos that I'm going to demo to you today. And I really would love it if some of you would go online to that website today and check it out. Going right back to where I was starting from, harmful algal bloom threats are not news in the Gulf of Maine. There's a long history of PSP in the Gulf of Maine. And now that I listened to this last talk, and it might not actually be PSP, which is paralytic shellfish poisoning caused by Alexandrium catenella. And some of the reports might just be a red tide caused by Mesodinium or Myrionecta. Mind blown. But we've been focusing on Alexandrium catenella and PSP blooms for a long time. Some of the data sets that we have like date back to the 1970s. And Alexandrium catenella has been present and blooming relentlessly summer after summer all over the Gulf of Maine. We've been focused on some of these places. So during the spring and summer, we usually have an IFCB in Nauset Marsh, somewhere close to Isle of Shoals, and Frenchman Bay and Bowdoin College. And in the fall and winter, we look at Pseudonychia and Dinophysis, which cause amnesiac shellfish poisoning, or ASP, and diuretic shellfish poisoning, or DSP. And so we move some of our installations to, I don't know how that happened. We move some of our installations to the URI campus, the Bowdoin campus, and Eastport in Maine. So because of climate change, we've been seeing a lot of changes in how these blooms occur, where they occur, and when they occur. So the long history of PSP continues. Alexandrium catenella has been present, continues to be present, but ASP and DSP are new threats in the Gulf of Maine. We have not before this seen Pseudonychia. I think the first time Pseudonychia was recorded was in 2014, and Dinophysis, although both both species of rhinoceros that we are concerned about have been recorded in the Gulf of Maine. The poison that they make, the toxin that they make, DSP, has not been recorded since before 2016. So there's a lot of new work going on all of this, especially because they cause aquaculture impacts and recreational impacts. And most of these impacts are now year-round instead of just being focused in spring and summer and fall and winter. So the sensors that we use to study these are the imaging flow cytobot, which you can see on the left, and the environmental sample processor. The imaging flow cytobot is my absolute favorite instrument, and we manage like 9 of them in the lab. I could talk about it all day. It's basically a combination of a microscope, a cell counter, a chlorophyll detector, a camera, and a computer that can be smashed into a can and put into water at 30 meters depth, or put on ships, or put on a dock. It's really, really amazing. And it can send data to you within 20 minutes of collecting it. And we've had some really great success with even very remote places like Alaska, where we collect the data and within 25 minutes, we can see what's in the water. The environmental sample processor is a similar instrument in a can, but it is more like a molecular biology lab in a can. It can analyze up to 60 samples because it has 60 pucks that can either do ELISAs or ERISAs, depending on which toxin you're looking at. And we have been using it to look at both PSP, so paralytic shellfish poisoning, and ASP, so amnesiac shellfish poisoning. And we've been focused on amnesiac shellfish poisoning over this one last year. So both these instruments do have limitations, though. The IFCV can only record particles that are between 5 and 150 microns in size. So right off the bat, I can tell you that it does not quantitatively record tripos, which we're interested in today. And the environmental sample processor, depending on which pucks you're using, can only analyze that kind of toxin, the kind of toxin that you have put the pucks in it for. And after you use up all 60 of them, you have to take the whole instrument out of the water. It weighs 250 pounds, and it is a pain in the neck to take in and out of the water. But the information that we get back from both of these systems are really amazing. So some of our Habon sensor installations that we did in 2023, this amazing video that we took of the Sea-Track boat, which is the unflippable boat, apparently, we have not been able to flip it, and it's been out in rough waters. We have a combination of onshore deployments of the IFCV and ESP, where in places like URI and Bowdoin College, we have it in their wet labs. And then in places like Frenchman's Bay, and in Nauset Marsh and in Lombo's Hole in Maine, we had it on these cool looking platforms, which have a little engine on them. This thing is extremely difficult to drive through Boston traffic. But once we get it out into the water, we can zip around in it and take it to wherever the bloom is. And then the instruments can sample in the water column, and we can control them remotely to sample the depth in the water column that we are most interested in. So it's a really flexible and amazing system. If anyone wants to volunteer to help me this summer, I will take any comers. And the third most interesting thing that we did last year was we deployed this Sea-Track boat for the very first time. This boat is as small as it looks. It's about 17 feet in length, and the IFCB sits sideways in it and samples from just the surface. So about 0.15 meters depth. And along with the IFCB, we also have a CTD in it, which takes other measurements. So this is also sending us all of the data live through satellite. And we can watch the boat. It's really cool. It also comes with a little remote control. So again, we are looking for PhD students. If you want to play with the robot boat, please come join our lab. So this was the first USV deployment that we did. So USV stands for Uncrewed Surface Vessel. It was nerve-wracking. The 5 days in May that we were kind of going in a big circle around the Martha's Vineyard Coastal Observatory, we were all taking shifts and up all night, just watching this boat and going like, please don't crash, please don't crash. In July, we did it for 32 days near the Isle of Shoals in Maine, close to Appledore Island. And we almost crashed the boat there once. And then ran away from us a second time, and we caught it. But what happened because of that is that we got a much wider data set than we had thought we would get. So, silver linings. So I just want to give you a short summary of all the different things that we saw last year. So PSP, or Alexandrium platinella, there was very little evidence of typical Alexandrium blooms along the Maine coast. So we didn't have much sighting of Alexandrium in Longboat Hole and Hartwell Sound. And there was no detection at all in Frenchman's Bay. But again, that may be because of factors like red tides that have been reported there may not have been Alexandrium, and that we got out there a little later than the Alexandrium season is. We were there from August to October of last year, and we would have really liked to have started there in June. But in Massachusetts, on the other hand, which kept us busy, there were record early and long blooms in North and March. So this Alexandrium bloom usually starts up in spring. So March 1st, we try to get out there and look at this bloom. Last year, the bloom was ripping from December 2022. It was a warm winter, and we think that's what caused it. And there was just so much Alexandrium in the water. And it was also toxic Alexandrium. So the peak shellfish toxicity was 10 times the norm at 5,000 milligrams per 100 grams of shellfish meat. So really the shellfish that was coming out of Nauset Marsh was completely inedible for the whole season. And when this bloom wrapped up, we also went and took some sediment samples, and there was record cyst deposition. So 8,500 cysts per cubic centimeter is about 2.5 times more than the number of cysts that we see deposited by these really big blooms every year anyway. So this was a bigger bloom than usual. The cyst deposition was also pretty huge. And so if we have a very warm spring this year, we might see another record bloom this year. So we are planning on going out there in February. Wish us luck. It is cold outside. There was also a very highly unusual closure of Pleasant Bay and Stage Harbor. So usually this bloom kind of stays up here in Nauset Marsh. This time it went all the way down here, and there was cyst deposition all the way down here. And all of these shellfish beds were closed from March through the end of summer, which impacted aquaculture quite a bit up there. ASP and DSP, so Pseudonychia and Dinophysis. Surprisingly, there were much lower concentrations of Pseudonychia detected than in the past few years. Like in 2021 and 2022, we had significant toxicity related to Pseudonychia up in Maine. It was being detected in mussels and clams. But this year, there was nothing. 2023, there was no toxicity detected in the clams. There were some particulate domoic acid detections in Harpswell, but they were just above the level of detection by the environmental sample processor. So it was present, but it wasn't really impacting the toxicity of shellfish meat. We also had an IFCB on the AZMP cruise, which went in the Canadian Atlantic. And this cruise, the main purpose of it was to see how Pseudonychia travels down from the Scotian Shelf into the Gulf of Maine. But again, the concentrations of Pseudonychia were just very, very low this year. DSP, so Dinophysis was present as usual in Nauset, but there was no toxicity detected. There was a really big bloom in Nauset Marsh, and there have been some surprising concentrations in December 2023. And when we went out 2 weeks ago, there were almost 800,000 cells of Dinophysis in Pleasant Bay. So that's a little bit south of Nauset Marsh. So we're kind of excited to see what it does this year and whether we'll be able to detect some toxicity there this year. And so we're coming to the thing that we're all interested in. So 2023, Tripos muelleri. I hope I'm pronouncing that properly. Apologies if I'm not. So Tripos species have been recorded in our IFCB records, but they were never dominant before 2023. So some of the other species that we had been recording were T. furcara, fusus, and lineatum. And I have example images from the IFCB of all of them. So T. muelleri has been kind of rare in our data set, so rare that we didn't have a classifier for it in our IFCB images. all of the images were just going to the Tripos class that we had, that was just like a catch-all class for everything Tripos related. So some of the data that I'm going to show from now on is very, very new. We made a new class. We had the classifier trained on some of the data that we just got from 2023. Also, another thing I want to repeat that I said before is that Tripos muelleri is really, really big. It's a distinctive looking cell, but the size range is between 120 to 200 microns. Also, I don't know if it's chain forming, but generally when phytoplankton bloom, they form chains, which are doublets or chains of one single thing. So if one single cell is 120 to 200 microns, then chains will be much, much larger and will not be able to make it through the intake mesh that we put on the IFCB so that the flow cell doesn't get clogged. The flow cell of the IFCB has limitations. So we try to put a mesh on it so that large cells don't come and just make a big ball of a clog in the flow cell. So this cell is too large to be sampled quantitatively by the IFCB. So even though we saw a lot of tripos, there were so many more, exponentially more in the water than the IFCB can ever record. So some good news is that the images of this class classify really, really well. So again, this is an initial assessment. We have only trained this classifier on the data that we got from our USV, so the Sea-Track boat data, which is 5 days in MVCO and 32 days near Appledore Island. But the precision and recall, as you can see in the plot on the left, so the precision is how good the classifier is at identifying Tripos muelleri positively. And the recall is if there's a large number of Tripos muelleri, how many of those can it positively identify? So the confidence level on that, the classifier score is close to 1, which is like 100%. And then the plot on the right is about the true positives, false positives, and false negatives. So the true positives has a confidence level of 95% on this classifier. So we are positively identifying Tripos muelleri correctly 95% of the time. There are a few false positives and false negatives, but they approach 0 at about 50% confidence level. So we can make this set a little better, and we will be training it a little further. With that, I want to show you how we've been using these data sets. So I'm going to go to a browser window and show you this website that we made. So this is the HabHub website, which ingests all the data that we get from the IFCB. Every single image that goes through the classifier and is trained to identify species is here. So this is a Tripos specific website that we made. So only the 3 Tripos species that the IFCB has really seen are in this. So Tripos muelleri is the purple triangle. So I'm just going to unclick this, and then we can only see the tripos. So one very exciting thing that I want to point out to you is that we added some spatial data sets. So this data set is from a cruise that UNH participated in last year. So some of the highest concentrations of key mulleri are from the ECOA cruise that some of you may have been a part of last year. So this data is from July 2022, and it is Tripos. These are some of the example images that this plot is showing you. So cell concentrations were at about 1,000 cells per liter in July of last year when this cruise was taking place. And you can click on this link and go to the IFCB data dashboard, which shows you every single cell that was in this data set that the IFCB collected. So this is one single sampling time point where we saw a bunch of Tripos. And this was on the ECOA cruise that took place last year. I think the next largest concentration of Tripos that we have is at the spot right here, which is from the Sea-Track deployment, which we had for 32 days from July through August. The highest concentration that the IFCB recorded was 7.5. So 7,500 cells per liter, which obviously is grossly undercounted. But most of the cells, most of the images that it identified are Tripos muelleri, it appears to me. I do not know what this is. Someone
It's just
else can-
sideways.
It is the same thing.
Yeah,
though?
it's just sideways.
Awesome. Okay, I'm feeling really good about this classifier now. This is a Tripos specific website, but if anyone is as interested in HABs as we are, we also have a habhub.hui.edu website, which has all the harmful algal bloom species that we are interested in. And this has all the data that every single IFCB that we have out there is making. And you can select as many species as you're interested in, and it will show you where those species were, how much the concentrations of those species were, and then you can go and look at the pictures and generate plots like this. So I can also go into this plot and deselect some of the things that I don't want to see. And then click on the ones that I am interested in. So this says that Pseudonychia has a 50,000 cell per liter concentration. And I'm like, wow, that's super high. So you can actually go and look at what the classifier is calling Pseudonychia here and verify for yourself that it was actually Pseudonychia. And then you can save this plot, you can download a plot that you're interested in, or you can download the CSV file and look at what data was actually ingested to make this plot for you. We want this website to be used by anyone who's interested in harmful algal blooms. It is open source. All of the data is there on the backend API. If any of you don't like to point and click, there is another link that I will be sharing by which you can just access the API of this website, query it, and get all of the raw data and do what you want with it. Publish it, make plots. Just, tell us what you're doing with it because we're so interested in seeing. So that's not the only data that we have on here. We also have shellfish toxicity data. And so you can select the data layers that you're interested in. We get shellfish toxicity data, shellfish bed closure data, and the seasonal shellfish bed closure data from the state. And then this pops out to tell you that this should not be used as a source of current information because we usually get information from the state after the beds have been closed. So don't use this website as like, oh, should I go shellfishing in this area? Please no. This is old information. We try to keep it as up to date as possible, but go to government websites if you're looking for shellfish closure data. But you can select that, you can deselect what you're not interested in and only keep the biovolume of the spatial grids or the cell concentrations of the fixed locations. So the spatial grid refers to cruise data. So the data that we get from all of the cruises that people do and have IFCBs on. And the fixed locations are the locations that we have at URI or Bowdoin College or our platforms. And if you are interested in seeing what that looks like, you can also just click on that and then go to the dashboard and have a look through the dashboard. So this is the dashboard that connects to our platform that we had in Frenchman's Day, and all of the data from the IFCB from 2021 current up to last year is here. So you can select the dates that you're interested in and go and look at singular time points if you want. It gives you information about the condition of the IFCB, what the settings were to the IFCB. You can also query this website and get the classifier information or the raw IFCB files. So all of the information that we collect, we want it to be open source, available for everyone, and as up to date as possible. So if you want to see what we've done with Tripos, the website is right here. Let me put the links that I have. Okay, I'll put them up after I've done the summary. I'm wrapping up, I promise. So to summarize everything that I will talk to you about today, 2023 was a very unusual year for HABs in the Gulf of Maine for us. Very low toxicity along the Maine coast, but very, very high toxicity and massive inshore blooms in Massachusetts and Cape Cod. Tripos mulleri was abundant across the western Gulf of Maine, and the northeast shelf in 2023. And Tripos was also present in the Eastern Gulf of Maine on the Scotian Shelf in 2022, which is a very interesting observation that we have for you guys to look at on the website. Quick acknowledgements. This is a very incomplete list of all of the people who have helped us gather all of this data and who continue to be really supportive of all of our efforts. I really want to point out that Mike Brosnahan and Ethan Andrews put in a lot of effort in this last 1 week to get the Tripos website up and running. The data is brand new. We've all been kind of pushing ourselves to get this data to you and get this classifier up and running. So I really hope this website is useful to people and that you guys are going to look at it. Here are some of the bookmarks. Please check them all out. There's a little tutorial for using the HabHub website. This link is to the GitHub. So if you even want to make one of the dashboards for yourself, you can find all of the information on there about how you can do that. And these are all of the IFCB data dashboards. If you want to look at IFCB images, if you want to take them, use them, please use them and just credit us for them. And that's my talk. I'll take questions if you have any.
Thank you so much. We're going to hold questions
Yes.
till the very end. So Dr. Thompson gave us the large scale hydrographics. We've got incredible data tools to work with. And I think take home was use the data, ask the questions, explore the data. This would be a good time for a workshop, maybe. Just have a workshop. Okay, so our next speaker, Dr. Liz Harvey. Let's see, I'll set you up here first. We'll be giving a little bit more of a species population dynamics talk, and
Okay.
you got it?
Spoilers. It's okay. Yes.
All right, and one last comment, stick around afterwards, first of all, just for the Q&A, because we're gonna be taking some questions and answers, and then afterwards, Martin Wozniak, are you in the house? Martin will be back and he'll be taking some tours of the high bay after we're done here, so take it away, Liz.
All right, thanks everyone. Thanks for sticking around to talk. We're gonna talk very much about the bloom, really go into depth with what we observed here at UNH and offshore. So we'll jump right in. First, before we do that though, there was a lot of people at UNH who were involved in this. I just got an email one day that was like, there's weird stuff in the water. Can you see what it is? And so I just want to acknowledge Doug, Kai, Chris, Sean, Sophie, and Sarah for all their hard work this past summer. This was, totally unfunded effort. And in terms of, we didn't have a grant or anything to do this. And so everyone kind of came together to sample. So I appreciate that. We also really want to thank the captain and crew of the RV Gulf Challenger who took Sean mostly out and Sophie out to collect samples. Diane Foster for her support. Sally Nelson, who helped convene today, and Opal and SMSOE for their support as well. So we're gonna back up a little bit. Maybe some people already know some of this information, but in case you don't, what the heck is Tripos? And I apologize in advance if I say seratium. Old habits die really hard. When I was a student, this was called seratium, so I'm still getting used to calling it Tripos. It is a dinoflagellate. Found worldwide and also very common to the Gulf of Maine, as we just kind of saw in the last two talks. As Maroon very nicely showed, there's lots of different species within this particular genus. Here are some images here. Of course, the one I outlined is covered here, but you can see other examples of the Tripos muelleri, the species we had this summer, and then there's various other Tripos species that we commonly see in samples all along in the Gulf of Maine. It is large, but there can be some smaller members of the species as well. Most of the time, though, we're seeing these really large cells. And I want to highlight that this particular group, most of them are what we call mixotrophic, meaning they can conduct photosynthesis, but they can also consume other organisms to get carbon and grow. And I think everyone who has been involved in sort of the bloom has their pet hypothesis as to why this bloom formed or how it was sustained. And one of my pet hypotheses has to do with mixotrophy. And so maybe we can talk about that after or something. Tripos is often found in the Gulf of Maine. This is going to be data now from the summer of 2021, not 2001, I think it says on there. But I was lucky enough to be out on a cruise right in Wilkinson Basin in June of 2021. And there was lots of serratium in the water. So this is just a graph from that cruise from the surface. About 2,000-ish cells per liter. This is lots of different species of serratium. So this wasn't just Tripos. Sorry, see, I said serratium. Damn it. Tripos. It wasn't just the species we had this summer. It was lots of different Tripos. And it was also found within a backdrop of lots of other species. So we actually had quite a few diatom chains that cruise. There was a lot of Sudnichia in the water at that point in time. So Tripos wasn't the only thing in the water, but we definitely saw it in, relatively significant abundance relative to, some of the other species that were there. The point is that this is not something brand new to the Gulf of Maine. It has been there before. And in case you still really don't believe me, this is a figure and data that was put together by the Massachusetts Water Resource Authority in a report that they released. And this is their counts all the way from 1992 to present of Tripos muelleri from their sampling areas. And so you can see that concentrations, I don't know if you can see that, but they alternate here, anywhere from 100 per liter but up to 10,000 per liter at some point in time too. But yet here are the counts and the observations from 2023, which are significantly higher than kind of the mean norm that we see. So I'm going to walk you through these different data sets that we have and we collected here at UNH, looking at concentrations over the course of the summer. And we'll sort of walk through each area. We're going to focus first here in coastal New Hampshire. We routinely sample weekly. I think lots of people do as well. They tell you when you get hired at a university never to start a time series, but I didn't listen to that. And so we have some of this data. What you can see at the top, I don't know if I can move this down. I don't know if that's at all helpful, but here we have time and the abundance of, see, it even says serratium here, Tripos, right here at our Coastal Marine Lab dock. We sample there once a week. And so you can see here in April, we have a very, very large peak of Tripos, almost 100,000 cells per liter. It sort of decreases, but we're still pretty high until we get to, I'm going to move this around, until we get down into August. So we had this sort of sustained population above 1,000, waffling in between 1,000 and maybe 20,000 cells per liter for pretty much the whole summer, starting in April, going all the way through August. And if I can make this work, that had some biogeochemical consequences. So the nice thing is that we also have some sensors at CML that take water temperature, salinity, oxygen, but really importantly, PCO2 concentrations. And so this is a graph that Chris Hunt put together for me, and you can see the sort of average in the background, sort of the, I don't know how many years, we can ask him later, but 20, thank you. 20-year average or 20-year data set on top of each other in gray, and then 2023 in red. And so what I want to point out is over here, this very low PCO2 concentration, particularly in April when we saw that very high peak, right, of Tripos. Everything else is relatively in line with its sort of 20-year average, but we did see these big low PCO2 values. Okay, so we're gonna move a little offshore now. This is out by the CO2 buoy. We call it, I don't know, I call it the CO2 buoy. The UNH runs a buoy that takes CO2 measurements along with temperature, salinity, oxygen, etc. We were lucky enough to get samples from there. And now this is looking at depth. So here's the surface of the water, and we would sample down to maybe 65 meters and cell abundance of Tripos. And you can see over here throughout May, June, and July, we had 10 to the 4, 10 to the 5 Tripos per liter that we were observing out here at the CO2 buoy. We did sample into August and September, and it was at that point that we saw a significant sort of decrease in cell abundance, or, you know, down to about 10 to the 2. Again, we could look at that buoy data and try to get at some of the biogeochemical consequences or implications of the bloom. And again, I want to highlight this very low PCO2. So it was low back at the CML samples, so back closer to the coast, but only for a very short period of time. Out here at the CO2 buoy, we had very low anomalous PCO2 concentrations throughout the summer as we had this very large bloom sort of raging on. And again, everything else looks relatively similar to the 20-year averages. Outside of there maybe is some excursions into low salinity. It's hard to know, you know, if those are real or, you know, if those are meaningful, I guess. So we see not only this high cell abundance, but we can also see the ramifications to carbon, CO2 concentrations in the water column because of this ginormous bloom. And so the question is, you know, what are then the ecological ramifications of those changes? And final data set that we have, we were able only very periodically throughout the summer to go all the way out to Wilkinson Basin. And so here are just a few samples from the summer, but it shows generally the same pattern, high, above 10,000 cells per liter in April, May, and June into July. And then we see that tailing off in August and September. And so this is really something that, you know, we observed all the way from very, you know, close to the coast. We sample at CML from a dock all the way out to a pretty deep basin in the Gulf of Maine. We see this sort of same repeated pattern of very high concentrations, April, May, June, July, with concentrations decreasing in August and September. I also wanted to show data from, we were very lucky to have NOAA taking samples for us on Ecomon Gulf of Maine cruise. And so we brought those back to the lab, we preserved them and brought them back to the lab to look at cell concentrations. And so here are some results from the first cruise that samples were taken on. This is in June. And it's probably hard to see the scale bar, but the highest value here is 20,000 cells per liter. And so you can kind of see, we had them sample to the south a bit in the mid-Atlantic bight, and then here in sort of the southern Gulf of Maine, this is surface concentrations and then concentrations of Tripos at the chlorophyll max. And yet you can see, you know, there are some very high areas, even higher than 20,000 cells per liter, and pretty high concentrations in the chlorophyll max as well at some locations. We were lucky enough to have the opportunity to get samples again from another cruise in August. And so by this time, again, the surface and chlorophyll max, this is on the same scale as the previous graphs. We only see a couple of really high areas of Tripos still sort of hanging around down here. And the majority of the Gulf of Maine is down in the thousands per liter, sort of a not abnormal concentration to see. So we were able to sort of get a pretty good view of how broad this bloom was, and that in combination with all the other reports from all of our friends that were emailing in observations kind of, you know, really started us figuring out that this was a much larger in terms of spatial scale bloom than we had ever anticipated. I also want to show this figure. Kim Hyde put this figure together and shared it with our email group chain. This is from June, showing the chlorophyll concentration in the Gulf of Maine and then the chlorophyll anomaly. And the two graphs on top are from 2023, and the two graphs on the bottom are from 2022. And so I think this is a really nice depiction of how intense, one, the bloom was, but two, how widespread it was, and lasted for a very, very long time. If you remember back a few slides when I went out in the summer of 2021, right, we saw a bunch of seratium and it was only over a couple of days, right? And so this is lasting months and months at a time. Okay, so the big question, or one of the big questions that I have, I don't know where to put this, is thinking about what sort of sparked the decline of the bloom, right? Why did it run out? There's sort of two or three main questions, right, you can ask about a bloom. Why did it start? Why did it keep going? And why did it stop? And so we were interested, and we thought we could at least try to answer the why did it stop question. So we did go out in October. We went and looked for, on the sort of sediment, fluffy sediment surface, for Tripos bodies. We thought maybe we could see them having sunk out from the water column and try to understand a bit more about what caused the bloom to decline. But when we looked at those sediment samples, we found lots of different cool diatoms, but zero evidence of any Tripos in those samples. However, also in October and November, my lab technician, Sarah, texted me like, she looks through all the harmful algal bloom samples for the state of New Hampshire. And she was like, I see a lot of dead Tripos in these samples. And so she took a bunch of pictures. And this is what we saw starting to wash up, both in our coastal New Hampshire weekly sampling sites, but also into Great Bay, the really large bay in New Hampshire here. These aggregates of dead seratium or dead Tripos bodies. We observed them through October and November. It's hard to see, but this is a really dense aggregate of just dead seratium bodies. And so it's really interesting to, one, think about where did they go from August, September into October? Are they, particularly sticky, causing these aggregations? And, what is the implication of them now coming back on shore and coming into the estuary? So I don't have any answers. I just think this is really interesting and cool. So we have a lot more work to do. We have samples from the summer that are still in process. Nutrient samples, carbon samples, HPLC samples. We have carbonate chemistry and eDNA that we're also working up. And we're hopefully going to be able to put that together and synthesize the story a bit better. But we're really happy to talk to anyone else who has samples and thinks they could be helpful in sort of piecing this story together. I think we don't have super great coverage for bloom initiation, right? We don't have a ton of samples from April into May, we have really good coverage from June, July, August, and into September. So we may be able to speak a bit about how the bloom declined. And these are just some of my outstanding questions, right? What initiated or fueled the bloom? How did it decline? And what was the fate of the material? And I'll leave you with some of, a couple of hypotheses I have. What was the role of mixotrophy in fueling this bloom? Was there any impact of grazers or viruses or parasites on the bloom's decline? And does Tripos have some unique aggregation properties, or maybe just their morphology, that causes these sort of large aggregates to form? And I'm happy to chat more. And I think now we finally have time for questions.
Yes.
I know.
All right, well, thank you so much. Maybe Maroon, you can also join us up front. Please. Maybe don't go too far. So we can have a little bit, maybe we can do 15 minutes of Q&A. Does that work all right for folks? And then Martin, you'll take folks on a tour to the high bay. So we will first take a question from online. Cameron, do you have a question you want to share from online? Then we'll take questions from the room.
Sure. So a couple of questions from Rena Karen and Jane Tucker, addressing terrestrial and riverine inputs to nutrients and fresh water into the Gulf of Maine, and thoughts on how might that have impacted the 2023 bloom. And I think these are directed to Dave Townsend. Could you hear me?
Dave, can you hear that question? And we'll give you first crack at it, and then we'll ask our other folks maybe if they can take a stab at it.
We might have to go back to him.
He's muted.
What's that?
They're muted.
Okay, now I'm unmuted, right?
Yeah.
We can hear you.
So like my students always say, I'm sorry, what was the question?
Sorry. So there's a few questions, sort of wondering what the influence of sort of river runoff, terrestrial fertilizer, sort of runoff from land in general, and just a very wet year that we've had, what that might have, how that might have influenced the nutrient properties this year, or just the general properties of the Gulf of Maine, and how that might then impact the bloom.
This is an issue that surfaces every now and then. Of course, we've been getting more precipitation, or at least more episodes of heavy rains over time. And the rate of precipitation falling on Maine, for example, if you fit a curve to it over the last 30, 40 years, it has increased slightly, but that doesn't influence waters other than falling on the surface of the ocean. And there are some dissolved nitrates in rainwater, but other than that, river water exiting Maine's rivers into the Gulf of Maine does not extend much farther than 20 miles offshore. It's subject to the Rossby radius of deformation. So as it flows downhill from higher sea level near shore in the rivers and estuaries into lower sea level offshore, it gets pulled to its right, and it forms this famous coastal current that Henry Bigelow described. And so, yeah, coastal waters will become fresher, especially farther downstream in the western Gulf of Maine and Bigelow Bight. But in terms of where we're seeing, well, for instance, the satellite pictures we just saw that Hyde shared with us are not, I don't believe they're influenced by freshwater discharges. We normally don't see a salinity signature that far off. So I don't think it was a factor in this bloom. But until I see a map of surface salinities, I can't swear to it.
Liz or Maroon, do you have anything to add to that?
I trust Dave.
You might want to, maybe you guys can move over here because I think the microphone is yours. Let's take a question from the room. Anyone have a question? Dr. Jones.
Liz?
Yeah.
So this is a mixotroph. What is it that they prey on?
Yeah, it's a good question. It eats, I think, in the literature, cryptophytes, small pico-eukaryotes. And so, on the very sort of large meta sense question, warmer temperatures is supposed to select for, sort of smaller pico-eukaryotic phytoplankton. And so maybe it's, I don't know, there may be some selection for that, which is then really helpful for these things that are mixotrophic. I have no evidence to support that other than the
What
hypothesis.
size are these? 5 microns or something?
Yeah, anywhere between 2 to 6, 7 microns, yeah.
Another question from the room. Yes, Betsy.
Yeah, so Dave said that nothing grazes on these things. Is he right about that? Do we trust Dave on that?
Yeah, so there's some evidence in the literature that copepods don't like grazing on tripos. It's really too big for a lot of microzooplankton, so they're kind of out. And so if the copepods aren't taking care of the tripos, then there's not, you know, not much, I mean, maybe some gelatinous zooplankton. I need
What do you think about parasites?
Yeah, so I'm a big on the parasite train. Tripos does have parasites that infect it, other dinoflagellate parasites that can infect it and take it out. It also has viruses that can come in and take it out as well. So I'm more on that, leaning toward that than being grazed down. I don't think many grazers can handle it.
Parasite train. We got 2 people on that train.
Anyone else is welcome.
That's not exclusive.
No.
Yes, question in the back.
Ratio beer bloom decrease PCO2. I just wonder mixotropy can decrease PCO2. So if it is not, it might be caused by photosynthetic stuff.
Yeah.
Just wonder your idea.
Yeah, it's a good question. I mean, certainly, I think
Can you repeat?
yeah, so the question is, if something was being mixotrophic, would that necessarily cause such a decrease in PCO2? Which would suggest potentially there being more photosynthetic. I don't know the answer to that. I think though that likely any mixotroph isn't doing one or the other in isolation. And so, you know, even if a mixotroph is getting a little boost from ingesting cells, potentially that has the potential to help, you know, help them sustain their populations.
We'll go to the virtual audience.
Yeah,
for the
we'll
next
go
question.
to an earlier question, just wondering about from Shelley Tomlinson. So you had Dinophysis blooms. Are these following Mycedium blooms? Is that,
Mycedium?
yeah, is that like a typical
No,
pattern?
actually, that's a question that one of our PhD students is investigating, but we haven't seen a lot of Mycedium at all, actually. And I was really surprised to hear Dave Townsend talk about the massive Mycedium blooms in Maine. I actually took pictures of the papers that he was sharing so I can give them to our PhD student and be like, here you go.
Yes, question in the back.
I was wondering, Liz, if you saw any hypoxia in your offshore cruises. I know there was some low DO.
Yeah.
From the MWRA data.
Yeah, that's the only hint that we have for low oxygen.
Was the
Yeah, was the MW data. We did measure oxygen on those cruises even in October. It wasn't particularly low. We haven't had any reports, but maybe there are other people who have observed something different, but we haven't.
Similar to what MWRA saw. In terms of getting down to around 150 micromole, something like that. And there was a large Ecomon cruise of the whole Gulf of Maine in August, and that oxygen looked okay.
Huh.
We were happy to see that it wasn't really dropping anywhere, so
Was the August cruise after?
yes.
Okay.
So there was a June cruise with good oxygen CTDs everywhere, and August everywhere. And it gives a good picture of the net effect, and it wasn't, I think the tide helps. Mixing in the Gulf of Maine is the tide is very useful.
Thank you.
Can you repeat that just for people online?
Yeah, so the question was about low oxygen and if we observed it, and Doug was remarking that there was oxygen taken on those Ecomon cruises, and there was one from August, and the oxygen looked okay, as Doug says. So nothing super worrisome in terms of being very low as a result of the bloom. And Doug states that the tide is very helpful in mixing.
We have about 5 more minutes. One more question in the back.
Have you done any culture work for Syracuse Tripos?
Yeah, I haven't. I wish I could have. We tried to get it in culture. It did not love being in culture. Yeah, so I wish. Maybe this summer I can get some. Yeah, Rob.
My question is about, what do we know about the biogeochemistry of tripos, and like, do they have any like uniquely high or low iron requirement, or do they have a high or low C to P, N to P? What do we know about makes them unique in their bottom up?
Yeah, that's a good question. I don't know, maybe someone, oh, sorry, the question was, what do we know about the stoichiometry of tripos? Do they have, or their nutrient needs, do they have a specific affinity to different nutrients? And I don't know the answer to that. Maybe someone else does, or maybe someone tomorrow will know more about that.
Diane?
Maroon, you mentioned that you have a particular screen on the outside of your flow sensor, which limits the size of these. And I would imagine, do they grow through the summer? So does that mean you're less able to detect them as they get larger? Or do they?
That's an interesting question. I think, so the question is that the IFCB has a size limiting screen on the intake, and would the serratium be more limited from going through the IFCB as they grow? I don't think we've actually seen that. All the tripos images that we have on the IFCB from years back, before this bloom was detected, are about the same size as the ones that we've detected during this bloom. And like I pointed out, we have images from the 2022 ECOA cruise. We have images from the EcoMon cruise. And we have some images from when we sent the IFCB on the Tioga around Cape Cod, and all of the images are about the same size.
And Liz, do you have the same limitation?
I don't. So I didn't talk about what instrument I have. I have something called a flow cam, which is similar to the IFCB, but a little bit different. And we have different flow cells that we can exchange. And so we can do everything from a millimeter, smaller. And so we're able to get quantitative tripos counts because we can, we're not gonna filter them out, pre-filter them out.
Cameron, did you have one more question from the?
Oh, I was going to just combine 2 questions. One was just generally, can you just describe the life cycle of a tripos? How long does it typically live? And then sort of what determines the size of the tripos? Is there any factors that size of the tripos?
That's a good question. Typically, I think tripos is probably like most dinoflagellates, and dividing every day and a half. It's a little bit bigger, so I think it's not, probably not dividing every day, but every day and a half-ish, I would assume. And things that limit their size, I think that's pretty species specific. Yeah, I don't know other significant things that might impact their size, maybe some nutrients, but generally, depending on the species, we have differences in size that way.
I wonder if we could just ask one more question of each of you. Many people may be coming into this just sort of having heard bits and pieces, like there was an event, there was something, tripos, we're learning about the species, we're learning about some of the hydrographics. How would you describe, what was sort of noteworthy or exceptional about what happened this past year? So why are we here, and how would you maybe just quickly describe that for someone who's interested in learning more? I guess I'd be interested in each of your responses to that.
Yeah, I guess for me, the intensity of the bloom, the duration of the bloom, and the spatial extent of the bloom are all novel in terms of anything that I've seen. And I've by no means, been in the Gulf of Maine forever. So there may have been other cases where, these large blooms happen. But it's rare that I've to see such a large, intense bloom over such a large spatial scale.
You have anything to add to that?
Yeah, what really stood out to us was how homogeneous the samples looked. So the IFCB, when it was sampling around the Isle of Shoals, Appledore Island, and even down near MVCO, there was only sericium and then a bunch of debris and nothing else. And I think when we've recorded tripos before, there have been other phytoplankton cells along with it, but this time there was nothing else. There was just tripos and then tiny things and debris that we couldn't really identify. And that's also really interesting because I think Dave was talking about this a little bit, about how Tripos and Alexandrium had been recorded in bloom form. I would really love to hear more about that. Like, why did that happen? How is it different now? It's really interesting.
So many questions. David, you want to take a stab at that and we'll rake?
Well, the short answer is I have no idea. The outstanding feature of this bloom that I heard about was from Walt Golette, who was working with tuna fishermen out of Portland. And he was constantly sending emails and texts and phone calls. And he never really made any measurements. The only measurement he made was, he said one day they killed a mola, an ocean sunfish. I didn't ask him why. And it flopped over on its side and it started to sink. And when it got 18 inches deep, it disappeared. And so the Secchi depth out there, well offshore, was 18 inches. And so the real question is, what did that do to the competing phytoplankton members in that community? And did it somehow reach such high proportions by getting to some sort of a threshold where it cut off the light for everything else and it could operate mixotrophically on its own and it just took over? And I think if we look hard for a specific cause in terms of a nutrient ratio or something along those lines, we're not likely to find it unless we build into our thinking these blooms happened in the past with other species. They're unusual events by definition. And so what might have been so unusual about this bloom this summer, we may never find out, but I'll bet it had something to do with light limitation, eliminating potential competitors, sort of a competitive exclusion principle in action. That's a wild guess.
Okay. All right, well, thank you. And thanks to all of our speakers. Thank you so much. You're all part of the research team now, so you've been informed. So let's try to answer some of these questions. Keep asking questions and talk to each other. Martin, you want to put your hand up? I think if you're interested in going on to a tour of the high bay, Martin will be